Hello, everyone. Welcome to our Albinism Awareness Series, a joint effort between the ILDS, the GLODERM Trainee Committee, and the Global Albinism Alliance. My name is Akash Varma. I'm the current chair of the GLODERM Trainee Committee, and our co-host today is Antoine Glickson, the executive director of the Global Albinism Alliance.
Welcome, everyone. So we will have 4 different sessions in this webinar series. We will first cover genetics, then dermatology, then we will have a webinar on ophthalmology, and finally, a last webinar addressing the social impacts of albinism.
And all of these webinars will have additional live Q&A time. So as I mentioned, your hosts today are me, Akash Varma, and Antoine Glickson. So I just want to give you a little information about the organizations which we represent. GLODERM is the International Alliance for Global Health Dermatology, and it's an official program of the International Foundation for Dermatology under the International League of Dermatologic Societies. The mission for GLODERM is to improve skin health for everyone everywhere by connecting dermatologists and trainees who are dedicated to improving skin health for all in resource-limited communities locally and globally. I represent the GLODERM Trainee Committee, and our main mission is to make sure that we disseminate information to individuals all over the world and increase education for dermatologist training in all settings.
And as mentioned before, my name is Antoine Glickson. I'm the executive director of the Global Albinism Alliance. The Global Albinism Alliance is a young international NGO that was launched about 4 years ago in 2020. Today, we represent about 250 associations and other NGOs that are serving the albinism community in all regions of the world. The mission of the GA is structured around 3 main pillars. We exist first to support albinism organizations that operate around the world. We act also to improve knowledge about albinism. And finally, we are the voice of the albinism community at the global level. And in that frame, we advocate to improve and protect the rights of persons with albinism at that level.
Great. So just to run over some etiquette for today's webinar, if you have any questions or comments, please submit them via the chat function. Our speakers will answer your questions at the end of the webinar. And we will be sharing further information about the GLODERM Trainee Committee and the Global Albinism Alliance in the webinar chat, so please be on the lookout for that.
Before we start, I'd like to remind you that this webinar series is put together and offered to you as a way to celebrate International Albinism Awareness Day, which has been celebrated since 2015 every year on June 13th. Our session today will cover one very important aspect of albinism, the genetics of our condition. As you may all know, albinism is a genetic condition. There are multiple genes that cause this condition and a very important number of genetic variants that can explain the existence of albinism in a given individual. We will have 2 different speakers who will address this specific topic. I'm now very honored to introduce Professor Benoît Arveiler, a professor in medical genetics at the Bordeaux University and University Hospital of Bordeaux in France. With over 15 years of dedicated research on albinism, Professor Arveiler has significantly advanced the understanding and diagnosis of this condition, including identifying key genes and investigating their regulatory elements and pathogenic variants. Over to you, Professor Arveiler.
My name is Benoît Arveiler. I'm a geneticist at University Hospital of Bordeaux in France, and I'm very happy to give you this talk about the genetics of albinism.
Albinism is a rare genetic disease that is autosomal recessive in all forms except ocular albinism type 1, which is X-linked. It is phenotypically heterogeneous at both the cutaneous and ocular levels, and it is commonly classified into oculocutaneous, ocular, and syndromic forms. Albinism is genetically heterogeneous with 20 genes, +1 for the related disease fonda. Key players are the pigmented cells melanocytes, which are found in the skin, in the hair bulb, and in the choroid in the eye, as well as retinal pigment epithelial cells in the retina. In pigmented cells, like here in melanocytes, melanogenesis is a complex process that takes place in specific organelles called melanosomes. Melanosomes undergo 4 stages of maturation from stage 1, which doesn't contain melanin, to stage 4, which is full of melanin, and these stage 4 melanocytes will be transferred to neighboring keratinocytes in order to protect the nucleus of these cells from UV irradiation. As can be seen on the left panel, melanogenesis starts with amino acid tyrosine and leads to the synthesis of both dark eumelanins and lighter pheomelanins. 3 enzymes are involved in this cascade, namely tyrosinase that orchestrates the first 2 steps as well as 2 steps at later stages, tyrosinase-related protein 1 and tyrosinase-related protein 2 or DCT. On the right panel, one can see other key players, namely OCA2, SLC45A2, and SLC24A5. These are ion channels which are anchored in the membrane of the melanosome. They regulate pH, and this is important because tyrosinase requires a neutral pH to be fully functional. We are now going through the phenotypic features of the different forms of albinism. Oculocutaneous albinism is characterized by hypopigmentation of the skin, hair, and iris, but it should be noted that this hypopigmentation is highly variable from one patient to the other, going from total absence of melanin to an almost normal pigmentation associated with more or less of a tendency to tan in some of the patients. Ocular features include nystagmus, foveal hypoplasia, misrouting of the optic nerve, iris transillumination, retinal hypopigmentation, all resulting in reduced visual acuity. Genes and proteins involved in these various forms of oculocutaneous albinism are melanogenic enzymes, tyrosinase, tyrosinase-related protein 1, tyrosinase-related protein 2 or DCT, proteins involved in melanosome iron and pH homeostasis, OCA2, SLC45A2, and SLC24A5, and one protein, LRMDA, which is involved in melanocyte differentiation and the processing of the protein p-mel. In ocular albinism, ocular features are the same as those described previously in OCA. Of note, there is no iris transillumination in fonda, and pigmentation is normal in these patients. Genes and proteins involved are GPR143, which is involved in OA1, X-linked. This is a receptor for L-DOPA, and SLC38A8 in fonda is a transporter for various amino acids in the central nervous system. Syndromic albinism is characterized by oculocutaneous albinism plus other clinical features. Patients with Hermansky-Pudlak syndrome, of which there are 11 types, have bleeding diathesis, reduced number or absence of platelet-dense granules, granulomatous colitis, and lung fibrosis. HPS subtypes are of variable severity. Chidiac-Higashi syndrome is characterized by recurrent infections, the presence of giant inclusions, neurodegeneration leading to progressive intellectual decline. Genes and protein involved in HPS participate in the complex process of melanosome formation and maturation and the transport of melanogenic cargo as shown on the figure on the right. As already mentioned, there is a large phenotypic overlap between the different forms of albinism. in such a way that just by looking at the patient, it is impossible to say which one has an oculocutaneous, an ocular, or a syndromic form of the disease. This is especially due to the high variability of hypopigmentation, but also it is possible that in some of the young patients especially, clinical features of a syndromic form are not apparent. Molecular analysis of the patients is therefore warranted. Let us now go through the genes involved in the different forms of albinism. Tyrosinase is a key melanogenic enzyme, variants of which are responsible for OCA1. OCA1 is the most frequent form of albinism, at least in Caucasians, accounting for about 40% of the patients. There are more than 500 pathogenic variants of this gene described in the literature. The phenotype of OCA1 varies from mild to severe, with severe patients having no pigmentation at all, like the one on the left, while the patient in the middle has some degree of pigmentation and the one on the right has normal pigmentation. This is due to the existence of mild or severe variants in the tyrosinase gene, the combination of which results in a phenotypic continuum. Let me spend some time on the thermosensitive hypomorphic variant C1205GA PR402Q. We have shown that this variant is associated in mild OCA phenotype in compound heterozygotes, characterized by a moderate to normal hypopigmentation with a tendency to tan in certain patients, and mild ocular features with a micronystagmus, low-grade foveal hypoplasia, and only moderately reduced visual acuity. It must be noted that R402Q is a frequent variant since it's present in 27% of Europeans, and that many individuals who are homozygotes for this variant do not have albinism. We therefore wanted to investigate further in which genotypic context this variant was pathogenic. For this, we collaborated with our colleagues in Manchester, UK, Dr. Seguniotis and Black, and we analyzed, as shown on the left panel, 3 frequent tyrosinase variants: R402Q in exon 4, S192Y in exon 1, and a promoter variant at position -301. As shown on the right panel, out of the 8 possible haplotypes, only 2 conferred a high risk of albinism. These are CAA in pink and CCA in green. These 2 haplotypes are rare, 1% for CAA, 0.3% for CCA in Europeans. These 2 haplotypes are considered as pathogenic alleles. Let us consider this pedigree in which patient 3-1 has a mild phenotype. She received from her mother a pathogenic variant displayed in blue with letter A, and she received from her father the pathogenic CAA haplotype displayed in red. This explains her mild phenotype. If we look at mother 2, she has the pathogenic variant in blue in the heterozygous state, and we see that at 402Q she has A, but in her A is associated with variant T and C at the promoter and at 192Y, and this TCA haplotype is not pathogenic. This does explain why the mother is not affected with albinism. If we look at the grandmother 1-2, we see that at 402Q she is homozygote for A, but on one allele A is associated with C and A in bold, and the CAA haplotype is one of the 2 pathogenic haplotypes. But on the other allele, A is associated with T and C, and TCA is not a pathogenic haplotype, thus explaining why the grandmother is not affected with albinism. In conclusion, Patients with these CAA or CCA haplotypes in the homozygous state or compound heterozygous with a rare tyrosinase pathogenic variant have a high risk of having albinism. It's generally going to be a mild form. These rare haplotypes are classified as pathogenic, and regarding variant R402Q, it has to be considered pathogenic only in the context of one of these haplotypes. This challenges the rare disease, rare and strongly pathogenic variant paradigm and raises the question as to whether other albinism genes may have such haplotypes. Coming to OCA2 now with the P gene and protein. As we already said, P protein is transmembrane chloride channel that regulates the melanosomal pH. OCA2 is the second most frequent form of albinism in Caucasians, 30%. It is associated generally with some degree of pigmentation, and there are more than 400 pathogenic variants described for this gene. Of note, there is a common exon 7 deletion that's present in African and African American patients and which represents 90% of alleles in these patients. Of note also, there are numerous rearrangements like deletions which are present in these genes and which should be looked for due to the high density of repeat elements in that genomic region. Considering the other OCA subtypes, OCA4 accounts for about 10% of albinism patients, but all the others are much rarer. X-linked ocular albinism represents about 7% of patients, is due to variants in GPR143, a receptor for L-DOPA. Being X-linked recessive, only males are affected, very rare females are affected, and heterozygous females have a mosaic eye fundus. Patients will have normal pigmentation. In Fonda, there is normal pigmentation, and as already mentioned, there is no iris transillumination. Hermansky-Pudlak syndrome represents between 6 and 7% of patients with albinism. Proteins involved in HPS assemble in 4 multi-subunit complexes, block 1, 2, 3, and adaptor protein 3 complex. Each of these complexes is involved in distinct steps of membrane trafficking and/or component sorting, which is required for the biogenesis of lysosome-related organelles to which melanosomes and platelet-dense granules belong. Chidiac-Higashi syndrome is a very rare but very severe and life-threatening disease. The function of the LIS protein involved in the syndrome remains poorly known. Defective LIS results in defective membrane targeting of the proteins present in secretory lysosomes. Techniques used for genetic testing of albinism nowadays include the following. First of all, next-generation sequencing allows to scan all 20 known albinism genes plus the Fonda genes simultaneously. This allows the identification of single nucleotide variants plus most of structural variants, including partial or complete deletions of genes. Additional techniques such as MLPA, QMPSF, or targeted array CGH can be performed in order to ensure the identification of deletions and duplications. Population-specific variants may be searched for with targeted approaches, and this is, for instance, the case for the OCA2 exon 7 deletion that is found commonly in Black Africans. Segregation analysis of the two parents must be performed in order to make sure that the two variants identified in the patients are in trans. Panel NGS commonly analyzes exons and near intronic regions and typically identifies coding and consensus splice site variants. But the sequencing of whole genes, including the exons, entire introns and flanking regions, allows to identify deep intronic variants that alter splicing, as well as variants affecting gene expression regulation. The use of all these molecular techniques provides about 70% diagnostic rate in the patients. So why perform genetic testing? First of all, having his or her precise diagnosis is right for the patient. Secondly, and most importantly, the main issue is to be able to distinguish between non-syndromic and syndromic forms of the disease. Then, having the exact genotype for the patient allows appropriate genetic counseling in terms of recurrence risk, 25% in most cases, 50% in boys in the case of X-linked OA1. In addition, prenatal diagnosis can be discussed depending upon the severity of the disease and familial history. In addition, the larger the number of patients tested, the better the knowledge about the disease in terms of genotype-phenotype correlations, the pathogenic status of certain variants, and questioning between monogenic recessive modes of inheritance and other modes of inheritance. Eventually, knowing the genotype may allow the choice of patients for clinical trial and may determine access to certain therapies when they will be available. As already mentioned, there is a large phenotypic overlap between the various albinism types. In this context, many patients are described clinically with so-called ocular albinism, and especially so in northern countries. But after molecular analysis, these patients may have, in fact, a variety of genotypes. Some of them may, of course, have OA1, but others may have OCA1, with especially the CAA and CCA haplotypes of tyrosinase. Other patients may have OCA7, FONDA, or even mild forms of Hermansky-Pudlak syndrome, especially HPS 3, 5, 6, and 8. And alternatively, some of these patients may have a differential diagnosis, for instance, with variants in FRMD7, an infantile nystagmus gene. The diagnostic rate of albinism is about 70%, which means that about 30% of patients remain unsolved. So where are the missing variants? Some of the variants may be located deep into the introns or in distant regulatory elements of the gene. Also, in the known genes, there are variants whose pathogenic status remains uncertain and which are therefore not considered as pathogenic so far. As already mentioned, there may be some differential diagnosis with variants in genes such as FRMD7, PAX6, CACNA1F, and congenital stationary night blindness genes. Albinism is nowadays considered a recessive disease, but it is possible that some alleles are dominant. And this may be eventually the case because some of the heterozygotes have a mild phenotype. Albinism is also considered a monogenic disease, but it is very well possible that some cases of digenism and oligogenism are existent. And of course, there may be more genes to be identified. To finish this talk, I would like to conclude with some messages. First of all, due to the extensive phenotypic overlap between the different forms, there's a specific need to test all 21 genes to establish the diagnosis. Second, I personally prefer the use of the general term albinism rather than distinguishing between oculocutaneous, ocular, and syndromic. Indeed, albinism corresponds to a phenotypic continuum, and the old classification may be misleading and give the false impression that the different forms can be distinguished clinically, which is not the case. This can be dramatic, since cases with HPS or CHS may be missed in kids, but also until adulthood. Third, albinism remains a largely underdiagnosed disease. This is mainly due to the existence of incomplete or atypical phenotype in patients, with some features being missing. foveal hypoplasia being of low grade, nystagmus not being present or not obvious, and mild hemorrhagic signs not drawing attention to a syndromic form, for instance. Due to the highly variable hypopigmentation in patients, the ocular features are central to the clinical diagnosis of albinism. These features must therefore be investigated with appropriate tools, and moderate and atypical signs must suggest a diagnosis of albinism. Finally, appropriate hematologic tests must be performed as soon as a syndromic form is suspected.
I'd now like to introduce our speaker, Dr. Robyn Kerr. We're excited to have her, a medical scientist and senior lecturer at the University of Witwatersrand in Johannesburg, South Africa, with a PhD focused on the genetic etiology of oculocutaneous albinism in sub-Saharan Africa. Dr. Kerr is deeply engaged in research and teaching, with a special interest in the ethics of genomic research in Africa. I'd now like to hand the mic over to Dr. Kerr.
Good afternoon. My name is Robyn Kerr. I'm from the Division of Human Genetics, the University of the Witwatersrand, which is in Johannesburg in South Africa. And due to the high prevalence of oculocutaneous albinism in our African population, our division has worked in research in both the genetic counseling field and in the molecular genetics field. We've worked on OCA for the last 5 decades. So I'd just like to share with you some of our findings and to let you know the status of OCA research and testing on the African continent today. So I'd just like to briefly discuss the prevalence of OCA worldwide, to remind ourselves about the basic inheritance pattern of albinism, and then to chat more specifically about testing for OCA in South Africa, which really is, as far as I know, the only testing, genetic testing, that is available on the continent. And then just to discuss some considerations about counseling and genetic services in general in low to middle income countries, and then to end with some thoughts on what we should realistically be doing on the African continent for individuals affected with albinism. So as you know, and discussed by the previous speaker, albinism is a group of disorders, and it occurs in all populations globally. There are different types of albinism, each caused by genetic variants at a different gene locus. And with respect to Africa, we now know that OCA1, caused by mutations in the tyrosinase gene, is essentially absent. There has only been one case ever written up in the literature of an individual affected with OCA type 1. OCA3 has been documented, but really it is well known that OCA type 2 is by far the most common form of albinism in individuals of Black African descent, and really this accounts for the high prevalence of OCA worldwide. So we now appreciate that the prevalence of OCA is not consistent through different populations in the world. In Europeans and in white population groups of European ancestry, the prevalence figures are around 1 in 15,000. However, in sub-Saharan Africa, OCA is one of the most common Mendelian disorders, and figures for South Africa are given at about 1 in 4,000 Black individuals being affected with OCA. And in certain other African countries like Tanzania, even higher prevalence figures have been reported. Also to note that there are certain population isolates across the world, like in Arizona, in the USA, other countries in the Central Americas, as well as some population isolates in Africa, where, due to the limited population size and the genetic founder effect, the prevalence of OCA is particularly high. So just a reminder on the inheritance pattern of oculocutaneous albinism. It is a classic autosomal recessive disorder caused by mutations in a single gene. So if you looked at a pedigree of a family with OCA, you would see the classic traits or characteristics of an autosomal recessive pedigree. Often skipping of generations, and often when an individual with albinism is born, their parents are usually unaffected. Just going back to some high school genetics, remembering that each of us has 2 copies of every autosomal gene in our genome. One of those copies we inherited from our mother, and the other copy we inherited from our father. So for an individual to be affected, they would both necessarily, that individual would have had to have inherited a copy of a mutated gene from each of their parents. So the parents of an affected individual are necessarily obligate carriers of the condition. In our context in Africa, this is a very important message to relay during genetic counseling. Because in African communities, superstition and myth surrounds the albinism condition, and it is mothers who are often blamed for the birth of a child with albinism, accused of doing something during pregnancy which resulted in an affected baby. So it is very important to remind families and communities that the parents of an affected individual are both carriers of a mutated allele. The second important point to emphasize during genetic counseling or community education around albinism is this fact that it is a biological condition caused simply by the inability of a cell to produce pigment. And again, in this way, we try to demystify albinism and to communicate the message that it is a medical condition and people with albinism are not different in any other way, and they do not have any superpowers. And we will revisit this discussion at the end of our talk. And as a reminder, if a couple has a child with albinism, then their risk of having a second child is 1 in 4 or 25% with each pregnancy. Okay, so as has been discussed by Professor Arveiler, in a first world diagnostic laboratory, extensive genetic testing is available. Due to the high prevalence of albinism in our population in sub-Saharan Africa, our division has undertaken research over the last 40 to 50 years. And at first, this research really addressed genetic counseling issues, but then later, when molecular testing became available, we set up testing for essentially the common mutation in our population. Okay. But in South Africa, working within a healthcare system with very limited resources, plus a healthcare system trying to cope with the huge burden of infectious diseases like HIV and TB, genetic testing within the state system needs to be highly selective due to the high cost of genetic testing. So early research found that there is a single common mutation in the OCA2 gene in Black African individuals. It accounts for nearly 80% of all disease-causing mutations in this population. And so during the late 1990s, when we were able to, genetic testing for this specific mutation was set up and offered via the state healthcare system. To my knowledge, there is no other diagnostic laboratory on the continent that offers genetic testing for oculocutaneous albinism. The test for this OCA2 mutation, which is a 2.7 kilobase deletion mutation within the gene itself, a very simple PCR-based assay has been designed, so the testing for this is relatively quick and relatively inexpensive. So this is the only diagnostic testing we offer, and this mutation, as we've said, but just to re-emphasize, is only a common mutation in the Black population. So if a European patient attends our genetic counseling clinic, there is actually no testing that can be offered to white individuals. If a Black family is found to carry, if both parents carry the 2.7 kb deletion mutation, then we are able to offer that family prenatal diagnosis if they request it. And as we've said, in our state healthcare system, there is no testing for any other mutations in this gene or any other. We have to appreciate that with limited resources, it is really not a priority to do diagnostic testing for oculocutaneous albinism. In the image shown here, one of our mothers, who is well known to our genetic counseling clinic, had non-identical twin babies, one affected with albinism, one not affected. And in an African population, the phenotype of albinism is so obvious that it really doesn't need even specialized clinical diagnosis, let alone genetic testing. So really, it is actually a misuse of state funds and healthcare funds to undertake genetic testing for simple diagnostic purposes. In cases where the mother or the family would request prenatal diagnosis, or in cases where individuals want carrier testing, then molecular testing is offered. In South Africa, we have a state healthcare system, but also a private healthcare system. And individuals who want comprehensive DNA analysis can have their DNA sent to an overseas laboratory to undertake such testing, as long as they can afford to pay for it. So really in the state healthcare system, the utility of testing, as said, would only be for really carrier testing and or prenatal diagnostics. But it is interesting to maybe pause at this point and just discuss the uptake or demand for prenatal testing as we have experienced it over the last few decades. So since 1999, when we could offer molecular testing for the 2.7 kb deletion mutation, we have had, or we have only performed 6 prenatal diagnostic tests in 5 families. And in most cases, the woman has indicated that the reason for asking for prenatal testing is simply to be prepared for the birth of an affected child, and only in 2 out of the 6 cases did the mother indicate that she would consider termination of pregnancy. So the issue of termination for an affected fetus is somewhat contentious, as the African community has differing ideas with regard to the severity of the phenotype. And this is really a subjective consideration, and different families have different views. But really, in a resource-constrained setting, unless a family wants prenatal testing, there is no particular value add for simple diagnostic molecular genetics. What are the more relevant issues that we have found that individuals affected with albinism and their families need on the ground? So in discussion with our genetic counselors, I've really gained some understanding of the issues that they experience at the coalface. So before we would want to consider extending our molecular offerings, what is needed really are people who can undertake genetic counseling. So genetic counselors or trained nurses or trained GPs who can actually speak the language, the native language of the population. This would be a far more practical and useful use of human resources in our setting. So the language divide provides an obstacle to true understanding and true communication in a genetic counseling situation. Further, we need to appreciate that global worldviews and cultural practices may differ significantly in Africa from a westernized and biomedically focused approach to practice, and also that there might be different religious views and different options available to people with respect to prenatal diagnosis and or genetic testing. So we really need to appreciate that genetics communication and services should be adaptable and sensitive to the context of the population we are serving. Just to consider quickly regarding this issue of accessibility to genetic counselors for people living in different regions of the world. In this paper by Abakan et al., 2 graphics really deliver the message very clearly. So if we look at the number of genetic counselors in different regions of the world, we can see immediately that there are very few genetic counselors, less than 50, on the entire African continent. The Royal College of Physicians in the UK recommends that 16 recommends that 6 to 12 genetic counselors per million is optimal to serve a population. So just to make the point again, that in numbers of regions of the world, less than one genetic counselor per million is actually functional in any given particular country. So then, how should we or could we make genetics accessible to populations like those in continental Africa? So we might suggest that we should train appropriate specialists who can speak the native language. So again, counselors or nurses often provide a very valuable service because they can speak the native tongue, and then they can be trained in the genetics so as to be able to communicate effectively with families. These healthcare workers could also not only educate families, but they could function as liaisons between the community and the actual healthcare system. So enabling individuals to get to the appropriate specialists, by way of example, when they needed ophthalmic care or skin cancer care. We should also really try and offer these services, get these services to the rural areas of our countries. And on a very basic level, we could produce pamphlets or posters educating the community and informing them about the availability of genetic services. So really what we are concerned primarily with in the genetic space is the management of ocular cutaneous albinism. So eyesight issues can be addressed by providing individuals with spectacles, sometimes contact lenses. Strabismus surgery is sometimes possible, often not. But really all individuals should be educated in terms of protecting their eyes by wearing wide-brimmed hats and protective sunglasses to reduce photophobia. Cancer is really the primary cause of death of individuals in Africa who are affected with albinism. So here, education again needs to come in the form of instructing individuals who are affected with albinism with how to protect their skin from the harmful UV that is particularly harsh on the African continent. Long sleeve clothing, hats again, and skin protection, sun protection cream if it is available to individuals. So with regards to skin cancer, here prevention is often definitely better than cure. Many individuals do not have access to dermatological and skin cancer treatment when such cancers arise. Lastly, I think it's really important when we are talking about albinism in Africa to mention the extreme problem that surrounds discrimination of individuals with albinism, the otherness of the phenotype as seen on the African continent. I would imagine that Iro will certainly talk to this again in the last session of the week, but just to set the scene somewhat. So in Africa, myth and superstition surrounds the birth of a baby with albinism and surrounds individuals who are affected with albinism. So affected individuals are seen to have some sort of magical powers. There are myths, including the famous myth that individuals with albinism do not die. In different countries, in different languages, there are words to describe people with albinism, and these often speak to the supernatural or to the spirit world. And unfortunately, what this has led to is individuals with albinism seeing as possessing supernatural powers. And unfortunately, this has led to some tremendous crimes against humanity. So individuals with albinism are maimed, sometimes even killed, for their body parts. Their hair may be cut off, arms, hands, limbs are cut off, and these body parts are then sold to witch doctors who make muti out of these body parts and sell it at very high prices to often businessmen or politicians who feel that these mutis then bring them particularly good luck. So what should we be doing? We should absolutely be educating communities as to the biological genetic nature of this disorder in a huge attempt to try and dispel the myths surrounding albinism and help individuals overcome this deadly discrimination that they are facing. So a particular NGO called Under the Same Sun has been doing some really practical, helpful things. They work in Tanzania, and they have been doing things by way of example, putting children with albinism through school in places where they are literally being threatened for their lives. They are housing these children in protective boarding schools so that they are being kept safe. Survivors of these kinds of attacks are being fitted with prosthetics or being linked up to overseas hospitals who are prepared to treat these children. And NGOs like this are also engaged with liaising with government institutions in forming national action plans with respect to albinism in these countries. So these are just some examples of the practical actions we can and should be taking on the ground. So just to conclude, we should really be training healthcare professionals who can counsel communities and families in the native languages. We should be providing rural communities with a genetic service. We should be engaging heavily in community education and dispelling the myth and legend surrounding individuals with albinism. And then I would say, lastly on that list, we should then think of strengthening and establishing genetic services for albinism, but only after the other has already been set in place. I really thank you for the opportunity that has been granted me to share my thoughts and share our African and particularly Southern African experience in terms of oculocutaneous albinism. Thank you so much for your attention.
Hello, everyone. As we are now in our Q&A session, please submit any questions you may have in the webinar chat, and all speakers will answer them shortly.
Oh, hi. Thank you very much, Dr. Kerr and Prof. Arveiler, for your interesting presentations and giving us the different perspectives from the global north and the Africa as well. So now we'll begin our Q&A sessions. So the first questions that I have is, how does the prevalence differ between different regions, and what factors contribute to these disparities?
So Benoît, please step in if you have anything further to say. But so OCA is the most common form of albinism on the African continent, and other forms are seen, and OCA1 not at all. Why this is a very interesting question, and actually we do not know the answer. We do not even know the answer to the question, why is OCA so common in Africa? Because if you think about the level of sunshine and UV radiation in Africa, you would almost expect from an evolutionary point of view that the genes for light skin would have been selected against in an African climate. But this is not what we see. And really, I have a very poor answer to your question, and the answer is we do not know why there's this disparity of prevalence of the different types, and also why OCA2 is actually common at all. It's counterintuitive, and I don't have a better answer than that. It's a very interesting question that we've been thinking about for years and really can't say. Besides the population isolates where we have people in the same tribe or living in the same geographical region, marrying each other, having children with each other, that's a genetic phenomenon with autosomal recessive diseases, that those diseases can become common in a particular place. But otherwise, I don't know. Benoît, do you have anything to
Yes, actually, hello everybody. I'm very pleased to be here with you. Can you hear me fine?
You are a little bit soft.
Is that better?
Otherwise, I don't know. Benoît, do you have anything to?
Yes. Can you hear me or?
Yep, we can, but it's a little bit of background noises.
Okay.
A little bit soft.
In fact, we have been studying. Yes. Can you hear me? Or? Yes, I can hear you fine, but apparently you can't hear me fine. I may try with. Is this any better?
Much better.
Is it good? Okay,
Yes.
great. I'll start again then. Oh, the problem is that I'm hearing now what I said half a minute ago, actually. It makes it a bit awkward. Okay. We have been studying 23 patients from. Okay, sorry. Is this any better now? I'm only on the Zoom session. Is that fine?
Yes.
You hear me? Okay. So I was trying to say that we have studied 23 patients from Mali, actually, and quite interestingly, we had a large majority of OCA2 patients, but none of them had the exon 7 deletion. And also we identified a few patients with OCA1 and with OCA4. So that means that these types of albinism are present in Africa. So, of course, I think at least in some populations, it will be important to look for these genes and not only for the OCA2 exon 7 deletion. This deletion, I think, is more present in the eastern, central, and southern part of Africa, but the rest of the continent may have other types of albinism. And in a couple of years ago, we collaborated with a colleague in Nigeria, and we had one patient with HPS1 actually. So, and I was about to ask you, Robyn, whether you had encountered some individuals who might have a syndromic form.
Since my time, we have not, but I would imagine that it occurs, but it's rare sometimes, unless patients present to the major hospitals. These patients would usually be missed by the healthcare system. So your rural clinics, we've never tested or come across these types of patients. But I would imagine that they're there. We just don't see them.
Thank you very much for answering the questions. My next questions that we have is, Dr. Kerr, you talked about stigma and discrimination specifically. Are there any community initiative or advocacy efforts aimed at supporting individuals with albinism in Africa? And how effective have this been?
So locally, we have albinism support groups who are very active in the community and support each other largely. When these killings, mutilations and killings of individuals with albinism started, especially in East Africa, then advocacy groups, we met one at a conference, the group Under the Same Sun, and I'm sure there are several others on the continent, but that one in particular is very well funded and supported, and they have done tremendously positive good in not only advocacy, but practically providing refuge for individuals who have been the victims of stigma and these so-called muti killings, providing safe haven boarding schools where the children can go and not fear. So I think advocacy is really an education of communities as to the cause of albinism and trying to get the message out there that it's a biological condition and individuals with albinism are not different. Their body parts do not have some mystical, muti. I think this is where we need to focus our attention at the moment. But I think advocacy is hugely effective, is and has been, as our support groups for individuals with any genetic condition. These groups provide a safe haven and an educational space.
Thank you. I think Dr. Jamila has question number 3 for you. Jamila, you might be on mute. You're able to come off mute. If not, I can help you with the next question. So the questions that we received is, thank you for the amazing presentation, Prof. Arveiler and Dr. Kerr. Can you please elaborate more on genetic counseling?
So I'm actually not a genetic counselor. I'm a scientist like BenoԻt. I'm just interested in the patient contact. BenoԻt, maybe you would actually be better posed to answer this question. I do not see patients mass.
You see the patients either.
No. Okay.
Basically, in our country, for instance, well, yes, of course, we have a whole panel of genes involved, as I mentioned. So it's important for the patients to know what form of albinism they have, and especially we have to look for the syndromic forms, which are, this is really the most important thing in terms of genetic counseling. Find out which patient has a syndromic form, because there are things that have to be done in terms of prevention regarding bleeding time feces, regarding possible problems at the gastrointestinal level and at the pulmonary level as well. These two things are not present in all patients, but they may happen in the most severe forms like HPS1, HPS2, and HPS4, for instance. And HPS1, as it happens, is the most common syndromic form. So yes, it's important to look for these ones. Then talking about, so that's the question about having the right diagnosis. Talking about recurrence risk, of course, then it's important to explain people and the parents what the risk is for next pregnancy, for instance. In terms of prenatal diagnosis, there are very, well, there is a very small demand for that. I mean, 1, 2 cases per year, but more than that. I think last year we didn't have a single request for that. So it's, people do not ask for prenatal diagnosis very often.
I'd echo that. I think that's what our genetic counselors do, to provide a definitive diagnosis is often very helpful to the patient, just that knowledge, and then explaining the inheritance pattern, how the genetics works, and risk to future offspring, and maybe other family members. That's usually the biggest focus of a genetic counseling session.
Yeah. And also, if I may add, one question we have in northern countries is that there may be, as I mentioned in my talk, there can be instances of individuals not having albinism but another disease. Then it's important to know what disease the patients have. Is it albinism? Is it something else? Then it's important because something else may not have the same consequences as only albinism, I would say. So it's important for the patients. It's important to know what disease they have, of course, because the recurrence risk may be different. Prenatal diagnosis may be more into question for other diseases, etc.
Thank you for the elaboration. So the next question says, as we do not have genetic testing in our limited resource area, what more can we do?
So I know in our context, when we speak to families or communities, the biggest message that we really need to get across is how individuals can protect their skin from developing skin cancer. So on the African continent, that's what individuals will die from. They will die from skin cancer. And there is this huge need to just educate parents, teachers in schools, and then as soon as the individual with albinism themselves is big enough, old enough to manage themselves. So sun protection cream is often very hard to come by, and even if it's available, it's very expensive. But ideally, we send the message that they would need to put sun protection cream, especially on their face and neck and chest. But then just to simply wear protective clothing, long sleeve shirts, trousers, big wide brimmed hats, and to try and avoid going out into the sun as far as is sensible. That is really the practical information that we try to give to families.
Thank you, Dr. Kerr. So the following question says, hey everyone, is there a way to prevent OCA in future pregnancies? Like, is there a way to even make any tests to the embryo before being injected when making in vitro? Thank you.
So the answer to that question is yes. If you have identified the mutations in both parents, you can perform pre-implantation genetic diagnosis as well as prenatal diagnosis for a pregnancy. But as both Benoît and us experience is that often there's little demand for prenatal diagnosis, but answer to that question is yes, you can do it as long as you know the mutations in each parent.
Thank you, Dr. Kerr. So we have the last question. How does one also obtain the posters showing the many races of PWA people from around the world?
I'm sure I understand the question, by the way.
They're asking about obtaining the posters. So how does one obtain the posters showing the many races of people with albinism from around the world?
Which posters are we talking about? We don't know. Really. These posters which are displayed and yes, displayed by the Global Albinism Alliance. So, I don't know.
I think it's from Dr. Kerr's presentation.
So if the question is referring to that early picture at the beginning of my talk, that is from the website of Under the Same Sun. So if they visited that website, they could certainly just simply download that image from Under the Same Sun. And I also showed at the end of my talk a pamphlet that our genetic counseling clinic gives to patients. And I'm very happy to share that if it was that they're asking for. But I think it might be the first poster they're talking to, which is off Under the Same website. The reference, I think, is at the bottom of the slide, but they can just find it online easily.
So I think that is the end of the questions we have. Thank you, Dr. Kerr. Thank you, Professor BenoԻt.
It was a pleasure.
Yes, back to you, Akash.
On behalf of GLODERM and the Global Albinism Alliance, we'd like to thank you for participating in our webinar, and we encourage you to go to our social media platforms to learn more about upcoming events, webinars, programs, and more. Anything to add, Antoine?
Well, thank you again for joining us today. We very much hope that you've extended your knowledge about albinism through this webinar session. And we very much hope that you will now share the information and at your turn raise awareness about albinism among your colleagues and in your location. Thank you very much.
And finally, I'd just like to thank our sponsors, Cerave, Ellis Foundation, and the International Society of Dermatology. Thank you all, and have a great rest of your week.
Thank you. Goodbye.